Hello, and welcome to episode number 3 of Antibodies, the official podcast of the Antibody Society. I'm your host, Jonathan Davis, founder of the consultancy Creative Antibodies. On this show, we plan to explore the science, stories, and people driving antibody discovery, engineering, and therapeutics with a mixture of personal stories and technical insights that we hope will give everyone a reason to listen in. Today, we are going to mix it up, do something different. I'm sitting at a table at the Antibody Engineering Therapeutics conference in San Diego, and we will have a series of guests sit down and talk with me about whatever antibody topic is of particular interest to them. You'll hear a number of different perspectives and personalities, and I hope you enjoy it. Okay, on with the show. Hello, I'd like to introduce Kat Harris, who will tell us who she is.
Yeah, so this is Kat Harris. I am Chief Development Officer at Rondo Therapeutics. I'm also going to be the president of the Antibody Society starting in January.
Which we're all looking forward to. So Kat would like to talk about target identification and new targets and how that relates to the surface of cells.
Yeah, so I think one of the themes that I've seen coming out of the talks so far here at the Antibody Engineering and Therapeutics conference, really started for me with hearing the keynote talk. So you had both Jim Wells as well as, Ray Deshays kind of highlighting these challenges in the field around identifying new targets, but also, specifically targets that are specific for example, tumor cells. And how do we as a field go after trying to identify those targets? And I think it's been a real challenge for the field to identify, for example, one target that has really high specificity for a given tumor type without being present also on normal tissue cells. And Ray Deshays explored that in his keynote to some degree, but again, I think that there aren't any really good answers out there for how we drive specificity to a particular tumor target, for example. DLL3 is an example he brought up as a target that is specific for small cell lung cancer, but it's a pretty unique target. And so I think this is a big challenge for the field.
Yeah, there aren't that many really clean targets, are there?
No. And I could think of maybe 3 or 4 that might have the potential to be, but a DLL3 is the one for solid tumors in particular that's been kind of showcased as an approved drug to have that specificity.
Well, one of the issues with specificity is that not only does it have to be specific to tumors, but it also has to be broadly present on all the tumor cells. To say a tumor is positive for some receptor, if that's true, but only 3/4 of the tumor is positive, you can kill 3/4 of the tumor. How much use is that?
Yeah, and I think the heterogeneity of the tumor microenvironment, which I think is what you're hitting at, is definitely another challenge around that. I mean, we know indications like ovarian cancer are notoriously heterogeneous when it comes to target expression. And that makes it even more challenging, you're right.
So one of the issues, I think, is, we are protein people and targets have traditionally been proteins. There are, of course, non-protein targets, and some of them can be extremely well saturated in the tumor. I'm thinking, there's a glycan that is on neuroblastoma cells. An antibody binds just that glycan, and it's found nowhere else except on other nerve cells, which can be a problem for side effects, but it's a very good target. It's called GD2, and I don't think there's a lot of neuroblastoma cells that don't have it on it. So we're starting to learn about other non-protein targets. They may be glycans, they may be other things, and I'm wondering what your thoughts are. Is that going to become an important part of our armamentarium? And do you think that combining, let's say, a bispecific that binds something that's not a protein and then binds something that is a protein might be a way to really specify the cancer cell and not have any escape?
Yeah, I think that's really intriguing. And I think Jessica Stark's talk this morning is kind of what you might be alluding to there with using the glycan receptor as one arm of the bispecific and then, a traditional antibody for the other arm. And I think the data there is intriguing, from a proof of concept perspective of going after a target like that. And GD2 was used in some of her proof of concept studies. I think we should be thinking outside the box for sure on non-protein targets. Like you said, I think they're potentially less well studied, but that doesn't mean that they wouldn't be good targets.
Right. The fact they're in a less well studied may very much be that they're harder to study.
Yeah.
Right? You can't look at the mRNA and say whether a particular glycan is being expressed or not.
Yeah. But there certainly are folks that are doing those studies.
And I think there's so much potential here. I was struck in that talk that she gave that there was a picture of the surface of the inside of a blood vessel, and the glycan structure is amazing. huge force. It's how can anything even ever get to the surface of the cell?
Yeah, I like how she described it as kind of decorating the cell, but at the same time, I don't even think that that really describes it. It looked more like a lawn or a forest, to use your word.
So since this is an audio podcast, I'll have to explain a little bit that Professor Stark is at MIT, and she gave this talk about looking at the sialic acid-coated glycans that tumors tend to produce, and showed a picture which showed just the inside edge of a blood vessel, and the glycan looked like a thick lawn of hay that was tall and tight and dense. And the point being that there's a lot of things there that you could bind to that tumors do differently. They sialate much more than normal glycans. So if that's something that all the tumor cells do, then that's a great target. And so I'm very interested to see how this works out.
Yeah. And I think there's been a number of just kind of thought-provoking talks like that today. I would say some of the other ones that have intrigued me kind of in that and binder idea of looking for, bispecific binders that you can engage to get better specificity or to get a desired response. Just heard Greg Lazar's talk on the Deletus platform where they're doing massive screening. Their technology and their binders are a bit of a mystery. They haven't really disclosed their platform publicly, but it'll be interesting to see more on that platform as they become a little bit more public with their data. But certainly this idea of going after the surfaceome, in these massive attempts to try to find combinations of binders that give specific activities, whether it's colocalization on a given cell type or a response when you combine these targets together is really intriguing to me.
I think a lot of this isn't very predictable either, so doing big screening is the way to find them. Although, of course, someone might come along and say, but AI could figure it out. So that was my last question for you. Do you think AI will play a big role in the future in coming up with the new targets or target pairs or approaches?
I think it's starting to have an impact, but I think it's guiding the data analysis, or you call this AI and ML enabled data sets, right? Where it is having an impact. But I think right now where it's working reliably well is a relatively small and contained area. I think that there's a lot of hope that that will continue to mature and have a bigger impact. Certainly, we heard Jo Viney's keynote talk as well. She's obviously a big believer in the machine learning and its ability to impact discovery and development of antibodies. I think that the proof's kind of in the pudding, and we're kind of still seeing that data emerge. Right now, it's still AI then verify with functional screening. So that verify with functional screening is still a very important step.
Absolutely. Yeah, we certainly aren't at the point where we can just get an answer, right?
Yeah, as much as, that would be nice to be there, I think there's still some work to be done in the field for sure.
So there's still jobs for scientists. That's good.
Still jobs for those of us that like our functional screens.
Well, thank you very much. It was a pleasure talking with you, Kat, and enjoy the rest of the conference. We're only halfway through at this point.
Yeah, great. Thank you so much for having me.
Our next guest is Bryan Glaser. Bryan is a friend and a former colleague from Invenra and will introduce himself.
Hi, I'm Bryan Glaser, Senior Vice President of Business Development at Invenra. Scientist by training, though. One of the scientific co-founders of the company about 14 years ago now.
Welcome. And what you've chosen to talk about today is bispecific antibodies, their role in the market, how it's growing, and how completely they will take over from monoclonals. And furthermore, will trispecifics come next?
That's right, yeah. Take over, we'll see. But we've always been intrigued with bispecifics, always fascinated by the potential to drive new biology that's not capable with monoclonal antibodies. From the earliest days at Invenra, we were very intrigued by the ability to empirically optimize affinity, geometry, epitope, and these multivariables in how they can drive new biology. And I think it's a huge opportunity first being explored in oncology, but I'm also very excited about opportunities outside of oncology.
So what are some of those opportunities? I've often seen people talk about using them, for instance, in autoimmunity, but there's so many disease areas. Are there possibilities outside of autoimmunity and oncology?
Yes, we've been starting to interact more and more with groups, say, in the virology space, where following some of the approaches from antimicrobials of multi-action, multiple targets to help prevent escape. There's also advantages with multispecifics there from a cost of good point of view, where you don't want to have to be developing multiple antibodies in combination. So I think there's some really interesting opportunities there. We've been starting to actually talk to people in programs in like heart disease, looking at different targets, probably with an inflammatory basis to it. So yeah, I think there's a lot of interesting opportunities outside of oncology once you start thinking about these programs at a mechanistic level and how can these antibodies really drive different biological responses.
So one of the things about antibodies or drugs in general is that our bodies tend to reuse things. So if you have a drug that binds a certain protein, it's probably in different places and may even have different roles in different places. So bispecifics can help there. And that might help with something like cardiology, where you want to hit only on the heart cells and not other places. So tell us how bispecifics can help.
Yeah, I mean, localization is a huge part of driving specificity and hopefully safety. And, bispecifics can provide, maybe one additional arm to locate to a new location. Trispecifics and beyond can even bring more of that power through AND gating and conditional binding. And so there's a lot of different tools when you start thinking about conditional binding, avidity, and ways to help localize biology to a specific tissue type.
So I just want to explain what AND gating means to our listeners. So yeah, AND gating, it's using the logic term AND or AND gate, which in logic is it is true if A and B are both true. And in this case, it means it will bind if target A and target B are both present, and otherwise it binds too weakly to do anything.
Correct. Yeah.
So that's a very powerful thing that bispecifics can do, because it can really help focus the molecules where you want them. So, okay, how much will this dominate? I mean, there's now bispecifics are starting to be approved for many indications, but still monoclonals are more common at the approval level. When are bispecifics going to be the most common approved drug in the biologics?
I mean, I think if you look at the development pipelines of a lot of larger organizations, bispecifics and multispecifics are starting to become a larger percentage of their early development pipeline. So you can see the interest from that early level. And now it's just a matter of them, being successful and being able to make it through these pipelines. And again, I think because they offer different mechanisms of action, there's more opportunities to really expand those pipelines. So how long it'll take, I don't know. I wish I could say, but I think you can see the evidence that they're definitely growing in proportion of the early discovery pipelines for a lot of organizations.
Now, Invenra has a new plug-and-play trispecific platform. Have you been getting a lot of interest in that?
Yeah, we're very excited about the platform. It's called the T-Body platform. It's been really built to try to have a true plug-and-play developable option for trispecifics, where the experience I hear from a lot of people that are trying to make, IgG-like, and you can use that term loosely at trispecifics, but things that have the properties of a strong IgG, a lot of these platforms have been custom engineering projects to just all the different chain pairing. And I think we have one of the first really robust solutions to enable 3 unique antibodies to be built into a molecule without scFvs or VHHs or common light chain. And so there's definitely interest. A lot of people are still very curious of, what are the advantages of a trispecific? What can a trispecific do that a bispecific can't do? And why would you want to use a bispecific? So a lot of our focus right now is trying to understand, and demonstrate different uses of a trispecific and what those advantages could be.
So as someone who's been in the bispecific world for decades, I can give a little history here that 20 years ago, the only thing people really thought bispecifics could do was to attract a T cell via CD3. And bispecifics were so hard to make that basically, if you looked at any review, it would start by saying bispecifics have great promise, but they're really hard to make. And that led to many ways of trying to solve that problem. Now that the problem is essentially solved with the Invenra technology and others, they've become really common. The same may happen with trispecifics. So back then, people were saying, what could bispecifics do aside from attract a T cell? Now, there are all these different ways of using them. And people are saying, what could a trispecific do? And I think moving faster, I think there's an acceleration, which I'm very glad to see.
Yeah, I agree. And I think what I've seen is it's going to follow a similar path where people are going to look at what has been successful from bispecifics and say, how do we take that next step forward? So a lot of people that we talk to are interested in trispecific cell engagers. There's been great work by Regeneron and others showing CD3 and CD28 paired with a tumor antigen or 2 tumor antigens with CD3. And so a lot of people are saying, are there advantages that trispecifics can provide over the T cell engager that you were mentioning for bispecifics? So an area where there's some understanding of the biology and what a bispecific can do, and can we take that next step forward? We're starting to look at trispecific ADCs, and I think there's a lot of questions. Do these molecules become too much? Do you have 3 binding arms and 2 payloads? And, at what point does the development path become way too challenging? And are there real advantages to doing it?
Yeah. But I could imagine a situation where you'd say it's a trispecific and any two of those three present on the cell will bind, and it just makes it much more difficult for a cancer cell to escape because it would have to eliminate two at the same time.
Yeah, I think that's one of the big advantages. We're also looking at using that third arm that may be specific for something in the tumor microenvironment that can cause, say, a conditional internalization. So another way to help with the safety aspect of the molecule.
Great. Well, we all are looking forward to seeing how things develop and really excited. I think it can only develop in a good way, that we don't really know what it's going to look like in 5 or 10 years, but there's going to be a lot of very good drugs, and a lot of diseases will have treatments that don't have good treatments now.
Yeah, I mean, I hope that the engineering solutions are no longer the challenge in these molecules. It is the biology. And so we now have the tools to build these molecules and address complex biology. That's my hope.
And AI comes along just in time to help us with that, right? We're not going down the AI route right now. So, well, thank you very much, Bryan. It's a pleasure talking with you.
Yeah, this was great. Thank you.
Bye. Our next guest is Basil Dahiyat. And Basil, tell us about yourself.
Hi, my name is Basil. I work at Xencor. I helped start the company about 28 years ago. We're protein engineers, and my background's in biophysics, and we focused on engineering antibodies, and in particular their structures and creating new modalities with novel antibody structures.
Thank you very much. That's great. So Basil says he would like to talk about the question of target discovery and are there new targets out there? So what is your basic thought about new targets and how to find them?
I think finding new targets is really more about generating molecule structures and modalities that can address those targets, right? Every target, I don't want to say every potential target is addressable, but I think you open up new universes when you create new modalities. I think bispecific antibodies opened up entirely new universes of targets. And as we elaborate on those, it's going to get better.
Absolutely. In some cases, I mean, a classic target for an antibody always had to have a biological reason to bind it. So you wanted to block it or maybe agonize it. With bispecifics or trispecifics, a target can be just an address. So that opens up a whole bunch of things that we don't know what they do, or that what they do isn't particularly important, but they can be addresses. Have you been working on those?
Absolutely. So our company's focused on, in the last decade, really building highly cytotoxic antibodies that engage T cells, T-cell engagers using mostly CD3, though also CD28 as the effector arm, and hitting tumor antigens on the other side. And what we've been doing is working on building structures that have that basic bispecific functionality, but that allow us to attack different targets than you could with first-generation technology. The distribution of a target is really, really critical. And depending on how you design your molecule, you can hit it selectively or not so selectively.
That's right. So not just the distribution of the target. Are you talking about on the cell?
They just be the target throughout the body on different cells, because there's no such thing as a tumor-specific target. They're just tumor-associated. They also live other places, and that's the hard part.
Right. So there's different ways of approaching that. There's using the tumor microenvironment. There's the masking, pH, anything like that, or using a second or third targeting arm to specify with an AND gate, which I've defined already for the listeners. So which do you prefer?
So we've focused on using the different level of expression on antigens between tumor cells and healthy cells and an avidity approach. So we just build highly stable modular structures that let us test, is one binding arm to the tumor antigen okay? Does that give you the selectivity you want versus healthy cells? In the case of a B-cell target like CD20, it's great. In the case of a renal cell carcinoma target like ENPP3, which is a program we have that's advancing rapidly in the clinic, it's also expressed on various healthy tissues that you want to avoid, but they're at lower levels. So we can use that old avidity trick, two domains, to tease it apart. And I think the next generation that I think is the AND gate. We're doing a lot of research on AND gates, and I think that's going to open up even more targets. We couldn't have hit ENPP3 before we built the modular structures to do 2+1 formats. I think there's going to be a whole new family of targets opened up by AND gates too.
Okay, so now I'm going to push a little beyond this. Targets don't have to be proteins.
Ooh, very true.
There's a lot of stuff on cells that is not protein, but that you can bind to, and it might not do much to bind to it. But if you get your antibody there, then you can have a T-cell engager or any other way of killing the cell. So what do you see as the future of non protein targets?
Well, I think the new characterization methods that are going to let us really look carefully at the structure. You could say, oh, there's glycans on the cell surface. Well, which one do you want to hit? Oh, I want to hit this particular GLICNAC chain. Yeah, but that's everywhere. But boy, as these people have these new mass spec tools and wonderful proteomics tools like we saw Jim Wells talk about earlier this week, you're going to have high quality information of subtle changes that we can then build antibodies against and say a glycan on the end of a protein chain, on a nucleo-based complex. That's going to be where the next world of selectivity might come. I hope to be retired by then, but we'll see.
That's right. Well, so me too. We'll be watching that from our beachside resort in some warm place, I'm sure. But
absolutely.
This is great. Well, thank you so much. I really appreciate talking to you and for your insights.
Thanks. It was a lot of fun, Jonathan.
Hey, my name is Jay. I'm a principal scientist at Johnson & Johnson. This is my third or fourth time here at the conference.
Okay, so I would like to pose your question that you would like to talk about. What do you think is the best way to evaluate developability of an antibody?
I think a lot of talks this year have focused on developability, and what we've known is over the past 5 to 10 years especially, the complexity of molecules has really increased, which brings with it a question of evaluating developability. With the infusion of AI and these high-throughput tools, I think we're looking at more molecules through the discovery pipeline, which then brings a whole new meaning of developability with increased complexity and more number of molecules. I think the field is still trying to figure out what the best way it is, and it's probably best to answer for that specific type of molecule, if it's a VHH or an SCFV or a FabFab.
Do you think that the suite of kind of half a dozen to a dozen assays that are typically offered by CROs is the way to go for this?
I think so. There was a talk, and her name escapes me, but the SVP from Merck provided a really nice overview of all the different tools that Merck is using. I'm not too familiar with the tools that are offered by the CROs, but what's been published previously in review articles and perspective articles, I think that's pretty solid. I think the challenge is still where within the pipeline to use them, because too much data is a problem and too little is also a problem at the same time.
Right. And you don't need to run every single assay to know if your molecule is good or not. So do you have a couple of favorites?
With the latest infusion of AI and machine learning, I think expression of molecules is key. Hydrophobicity, viscosity measurements. I sit more on the expression purification side of things, leaning towards manufacturability. Those are key assays that I look at. But of course, there's immunogenicity and all the other safety and efficacy measurements that also have to be thought about.
Okay. Well, very good. Thank you very much for your thoughts.
Yeah, thank you, Jonathan.
It's my pleasure now to introduce Shashi Jatiani, who will tell us about his work with FC effector functions.
Hi, Jonathan. Thanks for having me here. I'm from Boston, and the company is Seromic Systems. We are a CRO, and we care deeply about and are knowledgeable about FC effector function.
So FC effector functions, I want to start by giving people a little bit of a sense of what they are. They're part of the constant domains of antibodies, and they're functions that allow an antibody to kill a cell or allow an antibody to affect a cell in other ways. That's why they're called effectors. And they can be altered by mutations. This has been long known, but I think that Shashi has some new ideas about them.
Yeah, no, this is really cool. Thank you for telling us what an effector is. Effectors could be acellular, like the complement system and its components, because they bind to the FC. It could be a variety of cell types that express an FC receptor. And so each of these are effectors. And what we found recently, I mean, this has been important all along as part of regulatory filings where you have to look at complement activity, NK cell activity, phagocytic activity from mostly monocytes. But as complex formats emerge, I think this becomes even more important to look at it not just deeply physiologically, but broadly across all the effectors.
Okay, so what can you tell us about what you've been learning recently about effectors?
So there are two major themes in FC engineering. One is to enhance FC activity, and one is to silence FC activity. If we ignore the FcRn for now, which is mostly for PK and half-life, extension or reduction, and what we have been finding is there is no plug-and-play FC. You hope that every FC behaves the same way, but depending on the context, which I can elaborate on later, what are the various contexts which impact activity, you could have a silenced FC still retain function, or an enhanced FC not have function. And so that's what we're learning very empirically, just letting data speak to us.
So how does this happen? I mean, we've always thought of FCs as pretty much independent from the binding domains, the variable domains. Is there an interaction, or does it have to do with the geometry of the way it binds to a cell?
So all of the above. No, you're very cued in on this one, Jonathan. What we found is that it's just the way antibody discovery happens. You go through your campaign and get your binders first, and then you say, oh, FC, let's just attach what we want as an afterthought. But there is an interplay, and there's plenty of literature and data out there that shows that the Fab and the FC have not just interaction, they have interdependence. And we have solid data to actually show that is true. But to your point, antigen density can matter because it can cluster FC tails, so to speak, if the FC is the tail end of the antibody, and that'll elicit function where it wouldn't if it was not so dense. And then, you can have various other considerations. Overall, it's about geometry, is what the FC receptor on the immune cell that has, and what is density on the immune cell matters, right? So you could epitope distance from the membrane also matters. So sometimes if it's very proximal to the target cell, because of the proximity, you have elicitation of function, whereas a distal epitope may not have that degree of function or any of it at all.
So we have to think well beyond just target and the sequence of the FC we're looking at, but think about how is it interacting with its targets, how would it interact with itself, and do you also find issues with the Fabs bending back and interacting with their own FCs?
Yes, they do. So and you see this in an orthogonal readout, which is viscosity measurements. You have FC interactions, FC Fab interactions, and you have backfolding also. And when you look at that data, it gives you insight that this could be impacting not just in formulation context, it could be impacting in vivo activity, right? So for sure that matters.
Yeah, I think this is fascinating because it just shows that we always start out way too simple, thinking things are cartoons, and discover that there's much more to it than that, right? And so one thing that I've often been curious about is what you mentioned, viscosity. Viscosity goes up as there's more interaction between the molecules. And one question I have is how much of that is based on moments of dipole moments interacting with one another, rather than just protein-protein sort of normal kinds of van der Waals interactions or charge interactions?
Yeah, no, I'm not an expert on viscosity, but I do have a little background in protein structure. We know that these forces all matter. There are bonds, there are direct bonds, there are hydrogen bonds, there are van der Waals forces, there are ionic interactions. But I think it's the whole and the context, right? So not only is it a law of mass action, there's also an overall, to borrow a term, an avidity of all these bond interactions or side chain mediated interactions.
Okay, great. Can you just tell me, how did you get interested in antibodies in general and in effector functions?
For sure, yeah. My background is in small molecule oncology, so kinase inhibitors, transcription factor, blockers, and even agonists. But I've always had this desire to move towards larger molecules and biologics just because of the PK and the impact. At this time when I was transitioning, it was clear that antibodies were the future. And then the whole evolution of formats and specificities and engagements and context and just learning from other species, it was so exciting that I was compelled to make the transition.
Oh, wonderful. Well, we antibody people always love good converts. That's good. And you're also a singer. What do you like to sing? What's your what sort of things?
Yeah, I am multilingual, so I sing in Hindi, in Urdu, in English, and I'm a singer-songwriter. I write my own songs. I can't play many instruments except the trombone, so it's just thoughts that I have. I spend some time every morning thinking, and things inspire me. So most songs are born out of self-reflection, and then a tune will drop on me from somewhere, sometimes in a crazy context like driving or in the subway or in a shower, and then it becomes a song.
Yes, oh, that's great. That's what a lot of composers say, is that they just come out of nowhere.
Yeah.
Well, it's been a pleasure talking with you. Thank you very much for contributing.
Oh, I absolutely enjoyed my time with you, Jonathan. I hope people are listening and, we continue to interact and collaborate as a community.
Excellent. Okay, thanks. Our next guest is Brandon Dikoski, who wants to talk about our intracellular targets tractable. So, Brandon, tell us about yourself.
Hey, great to be here. I'm Brandon Dikoski. I'm associate professor at MIT and the Ragon Institute of MGH, Harvard, and MIT. And my lab works in high throughput antibody and T cell receptor screening. So we think a little bit about intracellular targets, and something that I think is at the forefront of kind of newly addressable targets that we can try to take a stab at.
So, what do you think is the most likely early use approach to doing this, and why do you think it will work?
So I think we're already working on this in a lot of ways, and I think that we're taking it on in the same way that our body naturally takes on intracellular targets, and that's through peptide MHC. So our T cell receptors are tuned and trained to recognize peptides presented by the major histocompatibility complex molecules. And there's already a number of antibody drugs that look at peptide MHC, sometimes in the context of neoantigens. And, there's TCR bispecifics, engineered TCR bispecifics, antibody-based bispecifics. And I think that's really the key. Obviously, there may be other ways where you can get inside the cells and activate an intracellular target specifically. But one easy way is to let the intracellular target come to you through the peptide MHC.
Okay, and just to help a listener who doesn't know all the letters here, an MHC is a major histocompatibility complex, and that is the molecule or complex that presents a peptide derived from some intracellular protein. It presents it to the immune system to see if the immune system wants to then kill that cell. So if you have a virus, it will present a piece of the virus. The immune system will say, boy, that cell's infected by a virus and will kill the cell. That is one way to go after intracellular targets, is let them become extracellular. There is an issue, which is that there's many different versions of MHC that different people have variations of. And that's important for our species, because if we all had exactly the same MHC, a virus could just mutate itself to not be seen by that MHC and it would not do anything. So we have many different MHCs among all the people. How can we have a drug that is useful to a broad population?
Yes, you're absolutely right. I think that's one of the biggest challenges in targeting these peptides presented by MHC is you've got a lot of heterogeneity. Different people are going to present different peptides from a certain protein based on their MHC presentation and processing pathways in the MHC proteins. So I think in the long run, that is going to be a challenge to address intracellular targets for every genetic population. That said, antibody discovery is getting faster, cheaper, easier, more effective. Developability is being addressed even earlier on. And so, I'm hopeful that instead of a small number of antibodies that we have in the short term, in the long term, there's going to be thousands of approved antibodies, where almost like a long menu where, pick your genetics, pick your targets, and there's an antibody for that. I think actually one of the big challenges that a lot of folks have talked about here this week is how do we get the clinical trials cost low enough so that these kinds of smaller fragmented markets can be addressed on a timeline and a cost that's feasible. But anyhow, I think that's one of the really exciting ways that we can address intracellular targets is by, again, letting those intracellular targets rise to the surface through the MHC proteins.
Great. Okay, now I'm going to ask you the real challenge, which is, how about addressing proteins when they're in the cell? And for instance, there's been a lot of talk recently about molecular glues, which are just molecules that can grab two different proteins and hold them together, kind of glue them together. And if one of those proteins is the type of protein that attaches something to another protein to tell the cell to destroy that protein, the molecular glue can be used to destroy a certain type of protein. Can we make things like that and express them inside cells and have them operate inside cells and still be called antibodies?
Maybe. I think we may be able to. Certainly, there's some exciting approaches, for example, using targeted LNP delivery, targeted nucleic acid delivery to specific cell types so that you can get expression of particular proteins based on external markers. And, we've got and OR gates for outside the cell. And so, maybe we can set up those kinds of and OR gates inside the cell too. There's a number of different mechanisms. I think they're a little bit farther afield than the peptide MHC-based targeting approaches, but I think we're going to get there.
Let's hope. That's fantastic. Okay, last question is more general. Is there anything that you've heard at this conference that really opened your eyes that you thought was a really interesting new thing?
Yeah, the whole week has been great. I think it's just been so exciting to see the iterations of bispecifics and multispecifics over the years and see how complex and how sophisticated they're getting, to look at different targets with different antigen expression densities and different functional activities, FC mediated or otherwise. It's just really a thrill to see all of that coming along.
Absolutely. Well, we'll be looking for you to continue to be up at the forefront of this at MIT, where you guys do so much great research, and it's academic research. You can publish it, so it's great. Thank you very much, Brandon. It's been a pleasure talking to you. Enjoy the rest of the conference.
Thanks so much, Jonathan. You too.
So I am Chris St. Georgi. I am CEO of a very small startup. Our business is nanobody discovery.
Welcome to the pod, Chris. It's a pleasure to have you here. And you would like to talk about what advances in antibody technology will be truly revolutionary. And I know you've developed your own technology that your company is using, and you have other ideas as well. So I'd like to hear what you think is going to be the next thing.
So the next thing is going to be, there will be an explosion of new therapeutics directed at previously impossible therapeutic targets. You're going to see this in part because antibodies are, in general, target proteins. And we know a hell of a lot about proteins now in terms of their genomics, with their sequences. We know how these sequences are associated with disease. But in the past, therapeutics were small molecules generated through very laborious techniques. And the kinds of targets to which you could bind small molecules were limited on the basis of chemical binding principles. Protein-protein interactions are universal, and they are easy to engineer and replicate using standard molecular biology and genetic engineering techniques. This kind of simple understanding, universal access to data, and screening platforms, it's all coming together. AI is the high energy fuel to all of this, because one aspect of recombinant engineering of proteins is you can tell the protein what sequences comprise. That's trivial from a scientific standpoint. And what's not trivial is analyzing I won't say billions of variants, because it's a much larger number. And in order to make sense of libraries and patterns, you've got to be able to bring to bear very heavy iron in terms of computational biology. And that's going to be revolutionary.
Yeah, no doubt. Being able to use AI to help us find the antibodies that are best is, of course, a big part of that. But you can also have lab techniques that help us find that. And are any of these really changing things right now?
So laboratory techniques have a much older history than the AI. It's generated over about the last 40, 50 years. So most laboratory techniques for generating antibodies are based on nature. And so you're either taking mammalian cells, or you're taking bacterial cells, or in the case of my little company, yeast cells, and you're asking them to produce these bits of proteins we call antibodies. You can also immunize an animal with an antigen, and the animal will produce antibodies for you. But the single antibody that the animal delivers is a highly screened and filtered result of a much larger repertoire of natural antibodies. And for therapeutic purposes, in the beginning, you want variety. You don't want a single antibody that the poor mouse or camel, in our case, decides is the best antibody for you. So you want to be able to have a large number of antibodies, and you want to have the way to analyze them. And if you apply synthetic design, so we can change the sequences, the number of sequences that we can change an antibody to are astronomical, okay, way beyond any practical screening technology or lab technique. So screening technologies can go up to a few billion. That sounds like a very large number to most people. And even to a scientist, it's a large number. But you need methods for analyzing data, patterns of sequences in proteins that are correlated with function. And you need to make better decisions on how to design the next library.
But do you have to use a funnel of some sort? I mean, if you have 10 to the 22 possible sequences or something like that, some big number, so much bigger than a billion or even a trillion, a computer is not going to be able to look at that either. It's too big. So how can we funnel it down and make it tractable?
So there is a concept called sequence space, which simply refers to the number of possible combinations of amino acids. And you have a linear sequence of amino acids that fold into a given protein. Sequence space is enormous, so you can't compute simply on the basis of enormous sequence space. You have to have other ways of approaching it. So you can simplify sequence space on the base of structural knowledge, which is highly complex and is developed over the course of the last half century. You can also simplify sequence space on the basis of pattern recognition. This is where AI comes in. So if you have data from screening where you've looked at let's say a billion sequences, there are certain patterns in there that the human brain cannot pull out, but AI and pattern recognition, machine learning is ideally suited to this. So this kind of information, a billion molecules with different sequences, that's called a training set. So you're training AI to recognize patterns. Once you recognize the patterns, you put them into your new library and come up with a new set of variants, and you have another training set. So AI output gets better, more intelligent, if you want to use that term, and our ability to isolate molecules that have the desired functions increases enormously. And we're just at the very beginning of this. And right now, what's happening is you have a tremendous amount of interest and hope in designing antibodies from scratch, de novo design, but in part because of these sequence space issues, that's really not easy to do and probably not possible yet. So you need feedback from actual screening, actual biological experiments in order to train the AI to get there.
Well, thank you very much, Chris. It's been a pleasure talking with you, and good luck with your new company and trying to revolutionize biology.
Well, thank you very much. It's been a pleasure talking to you, Jonathan, and my best wishes for you and your enterprise.
Thanks very much. Bye-bye.
That's all for today's episode of Antibodies. Thank you very much for listening. I hope you can join us for our next episode. Meanwhile, check out all the materials and announcements relating to antibodies, including information about our sister podcasts, at the website antibodysociety.org. The executive producers of Antibodies are Kristen McPike and Josh Dorai, with additional production and ideation help from the many volunteer members of the Meetings Committee of the Antibody Society. Editing and original theme music is by Jeremiah Davis. Antibodies is a production of the Antibody Society.